On a bit of a more positive note, though, omega-3s have a bit more scientific backing. There's some more research into them. They seem to only do good things for the body, which is nice for us. We know that omega-3s are required for normal brain development. So in development through the womb, these are required. If we look into Alzheimer's patients, we notice that N3s are reduced in the brains of Alzheimer's patients, and specifically DHA is reduced in the brains of Alzheimer's patients. So the next logical thing to ask is, if we give omega-3s, can that correct Alzheimer's disease? Or at least if we give DHA specifically, or maybe all omega-3s, can we delay the onset of Alzheimer's disease or help improve the negative effects of Alzheimer's disease? Now, in humans, we have some whole body evidence. We don't really know much of the mechanisms, again, because brain research is so hard to do. This is in older people. This is 6 months of supplementation, either with a control or with an omega-3 fatty acid. And then for the following 6 months, from 6 to 12, everybody got omega-3s. Ethically, it seems like this is the right thing to do because we're inclined to think omega-3s are good for you. So it wouldn't be ethically reasonable to just give people a control diet. And you can see in these graphs here, the black lines, the black circles, are those subjects that got the omega-3 for all 12 months. The open circles are those that got the control and then the omega-3s. And these graphs are showing scores on that MMSE test that we talked about before, and then the two panels in the middle and the right panel are specific areas of emphasis within that test. So looking specifically at word recall or attention in the middle and the right hand graphs respectively. So these are components of the MMSE. What you'll notice is that in each case there's a decline in MMSE score, or cognitive function, that is less pronounced with omega-3s. And not only that, but when the control group was transitioned to the omega-3s, the declines that they were seeing seemed to be less severe. And it's a little bit hard to judge that in the first two, but certainly in the third panel, the one with attention, as soon as the open circle control group switched to omega-3s, there's really a reversal in the attention score on that MMSE test. So this tells us that omega-3s are good at least preserving normal cognitive function. The decline is not as abrupt. Why? Why does that happen? We don't know in humans, and we have to go to a model where we can investigate a bit further into the brain. So these two slides were reversed. That one normally followed this slide, but this is the mechanistic slide looking at transgenic mice that have a tendency to develop those beta amyloid proteins and plaques. As a result of that plaque formation, they develop Alzheimer's disease. And so using this model, we can investigate whether omega-3 fatty acids either affect Alzheimer's disease at the whole body level, or maybe they affect plaque formation or the different forms of beta amyloid proteins. What we see is when we separate into a control group, a group that's given low DHA, which really is none, this treatment is meant to deplete DHA in the body. Even in the control group, they got a maintenance dose, or the food that the control group ate had some DHA in it, but when given none, it apparently depletes the normal DHA that these mice have in their bodies. And then a high DHA group that were given 0.6% of fat as DHA, which if you extrapolate that to humans with an 80 gram fat per day diet, that's around 500 milligrams, which isn't much more than maybe 2 or 3 fish pills, depending on what kind of pill you take. What we see is that when we give DHA, it reduces the beta amyloid 42 protein. This is the bad one. the really long one. So we can see on the left-hand side, this is the beta-amyloid 42 protein in the control and the low DHA group. There's the same amount in the tissues of these transgenic mice. When you give high DHA, that goes down for some reason, which is really good because that would lead us to believe that there should be a decrease in plaque formation and there should be a decrease in Alzheimer's. This jives with what we know from the human data. Not only that, but there's a decrease in plaque formation of 40%. This isn't represented here in this graph, but when looking at plaques in the brain, that also goes down significantly with DHA supplementation. Now, what's kind of interesting on the right-hand side is in the control and the high DHA groups, we don't really get a change in the amount of the normal length beta-amyloid protein. But for some reason, when DHA is reduced on that low DHA diet, the shorter beta-amyloid protein goes up. We don't know what the effects are. We don't know if that increases plaque formation. We don't know if that causes more Alzheimer's disease. But it's likely that that's undesirable. You wouldn't want that to happen. There's still the chance that those could accumulate and cause plaque formation. So pretty interesting. Along with the effects of amyloid beta proteins, N3s may improve Alzheimer's disease through some other studies have shown a decrease in inflammation, things like TNF-alpha in the brain. And there's also been studies looking at cerebral blood flow and oxygen delivery, and that's been linked to improved brain function as well. So there's no clear-cut reason why yet, but omega-3s overall seem to be fairly good. One thing to consider is that both fish oil and fish oil supplements do this. It doesn't matter if you eat fish or if you take the supplement form of fish. They are good at reversing the amyloid beta plaque accumulation. And on a whole body level, they reduce cognitive decline, but there's no agreement on if they reduce Alzheimer's disease. Okay, that's an important distinction to make. Cognitive decline is part of Alzheimer's disease, and again, it's a fairly nebulous term. It doesn't really define, or it's hard to define, but tests of cognitive decline don't show as much of a decline with fish oil. But some show beneficial effects for Alzheimer's, some don't, so it's still controversial at this point. Overall, I had a few thoughts just on this issue, the fat picture. Coconut oil, starvation, omega-3s. And in my mind, it seems as though if the brain is starved, which is what we were talking about in the coconut oil video, shouldn't we be able to measure a change in VO2? If the brain isn't getting enough energy, the brain works aerobically and exclusively uses carbohydrates or ketones, which is a carbohydrate analog that we make from fat. Shouldn't we be able to measure a decrease in metabolic rate if that's the case? And then if we gave coconut oil, or if we gave omega-3s, that would restore the normal supply of substrate and allow a normalizing of VO2 or an increase in VO2. It seems like that would be a fairly easy way to measure this, and we might be able to do this in people. It would be a bit technically difficult because you would have to do the AV difference between the carotid and jugular vasculature, which isn't something that you want to mess with, but it could, in theory, be done. Now, it's possible that there's a role for medium chains in this picture, depending on where the blockage is. If it's that substrate can't be taken up into the cell, then medium chain triglycerides can bypass that and slip right through. If signaling is being interrupted somehow, then medium chains don't require signaling. They can get right through. But the downside, the other side of this argument, is that they could just accumulate as well in the tissue. If for some reason metabolic rate is depressed and it's not because of a decreased fuel supply, then these medium chain triglycerides wouldn't be used, and they would just sit there accumulating, and we don't know exactly what effect that would have. Omega-3s, on the other hand, like we've thought of a lot before, they could improve membrane fluidity for neuron cells and other brain cells. Maybe that helps improve signaling. Maybe that helps improve processing the amyloid precursor proteins. Maybe that helps transporter function, getting carbohydrate or ketones into the cell. Or maybe the omega-3s have that other classic action of activating PPARs and PGC1 alpha, increasing mitochondrial content, and maybe clearing out whatever fat or other byproducts are accumulating in the cells, if that's what the problem is. These were the first things that I thought when we looked at this fat picture, and then I immediately thought, this is everything we've talked about forever in muscle and adipose, and it really strikes me that this is familiar thinking. It's a really good place to start, but the brain in Alzheimer's disease might not be the same thing as insulin resistance. We might need a pretty drastic reassessment of these mechanisms and the way we think about these problems in order to generate any real insight into Alzheimer's disease. I think it's a good place to start, but I think it's dangerous to always think in the same manner about these things, just in case you develop these biases. We might need to approach this problem from scratch. Now let's move on to brain games. I don't want this to run too long. It's going to end up being a full lecture, but let's see if we can get through brain games and some other stuff. The main goal of brain games is to achieve what's called far transfer. In the brain game neuroscience area, far transfer is transferring of the improvements that we observe with games to real-world situations. Something that is a non-game skill. And this is really difficult to quantify because all of these games have built-in metrics, and life doesn't necessarily have a built-in metric. We need to establish some kind of test that mimics real-world problem-solving situations and real-world decision-making and attention and focus situations in order to know if these effects transfer. In general, brain games that involve many different components of cognitive function, these complex games, seem to have beneficial effects over simple games. So things that combine attributes and don't just focus on single things like attention or memory or speed. Things that test problem solving, task switching. These things require us to take in information and process it and have an appropriate output, and to do this with a lot of different individual attributes. So we might need speed and memory and attention to solve a problem. Now, one test that's been developed that tries to address this issue of far transfer is called the Everyday Problem Solving Test, which is meant to mimic things like choosing groceries in a grocery store, differentiating between labels of products. It's right now the best estimation that we have of the ability of games to affect real life. And in this study, this is a fascinating study. I was listening to a podcast while preparing this class earlier this semester and heard of these games being used, which are Brain Fitness is a Lumosity type game. Rise of Nations is an off-the-shelf computer strategy game. And then Space Fortress is somewhere in the middle. It's not quite strategy, but it's not a Lumosity type game. And so this is just recent research. I contacted the researcher to find out more about this stuff, and she was nice enough to send this information back. Here we can see that different games have different real-world effects. So for some reason, Rise of Nations, which is a strategy game, didn't affect the everyday problem-solving test. Here we're looking at the amount of time to solve problems. And this is the change from baseline to after 6 weeks of training, and so a lower time is better. You want to be able to solve these problems faster. There's no difference in the Rise of Nation game, but Brain Fitness and Space Fortress both seem to improve everyday problem solving, which is exactly the point of these types of games. So pretty exciting stuff. We don't know exactly why this is the case yet, but it's really neat to see real-world changes from these games. Now I want to move on to the next topic and show you a clip of how calorie restriction can affect cognitive function. But both BBC and YouTube didn't like that and removed the video. So instead of including that video in this video, I'm including links in the description that will point you to the documentary in question. So if you go to the description and click the video 2 link, that'll take you to this documentary, and then I want you guys to watch from 43 minutes to 47 minutes 53 seconds. So go ahead and do that now, and then resume this video when that's done. So that video is pretty impressive in some of the claims that it makes, that you can prevent the development of Alzheimer's, at least in mice, the equivalent of 30 human years. And the opposite too, you can accelerate it by 15, 20 years by eating a high-fat fast food diet with lots of fructose. So the research that spawned those claims is what we're looking at next. This is Mark Mattson's study that he's referring to in that video. He's using a transgenic mouse, like they said, that develops Alzheimer's. There's 3 mutations, and those aren't specifically important, but that's the 3XTGAD. That's what that means. There's triple transgenes in this Alzheimer's disease mouse. They end up developing those beta amyloid plaques. They develop the neurofibrillary tangles. It results in Alzheimer's, and they've reduced cognitive function. So what happens? Creatine, calorie restriction, but not intermittent fasting, reduces the beta amyloid plaques. And you can see that here. If you guys were paying attention during the video, right near the end, you'll notice that he said that intermittent fasting seemed to be better than daily calorie restriction. So initially, this result seems to go against that. What we're looking at here is the amount of the beta amyloid protein, both the 40 amino acid and the 42 amino acid form. You can see that when these transgenic mice were just left to eat whatever they want to, they had a certain baseline level that decreased with daily calorie restriction, but it was essentially unchanged with intermittent fasting. Intermittent fasting here is equivalent to the alternate day fasting that we investigated last semester. So we get a beneficial decrease in these beta amyloid proteins with only calorie restriction. No change seems to happen with intermittent fasting. So not a great case for intermittent fasting here, and it goes against what he said in the video, but if we look at the whole body, the whole mouse cognitive performance data, and this is just in females from that same study, we can see that there's something else going on. There has to be something else going on. We notice here that both of the diets improved maze performance. So what we're looking at on the top panel is the amount of time that it takes to complete the maze. So a shorter time is better. And at the bottom is the path length that the mice took to complete the maze. So a shorter path length means that they're more efficient at finding and completing the maze. Now the light pink line in both cases is the transgenic mouse with Alzheimer's disease. It took them longer to finish the maze, and they walked around further. So they were poking around the maze, and you saw that maze in the video. It was the star-shaped maze with the reward at one end with all the arms coming off the center piece. That's what we're talking about in this graph. And then you'll notice that in both non-transgenic animals and the 2 groups that calorie restricted, regardless of whether it was calorie restriction on a daily basis or intermittent fasting, the time to complete the maze and the path length was reduced in those cases. And this is pretty important. Both calorie restriction and intermittent fasting restored normal performance, back down to normal. So the non-transgenic, non-Alzheimer's disease time and path length was matched by both groups. Now we saw this in males and in females, so it's not sex specific. One question that I have though is maize performance really cognitive function? How do you translate this into thinking or attention or focus in mice, let alone in humans? And so this is interesting. We see an improvement in performance in the intermittent fasting group, but we didn't see a change in the mechanism, the amyloid beta proteins on the last slide. So this suggests that somehow intermittent fasting can protect against the negative effects of amyloid beta proteins. They're still there, they're still exerting what we think are negative effects, but somehow intermittent fasting protects against them. And maybe it has to do with developing new neurons like we saw in the video, but we're not entirely sure why at this point. Altogether, I find this area to be a fascinating area of study, how the brain changes and that omega-3s can modify that. Calorie restriction, which is such a weird intervention on the topic of lifespan and Alzheimer's disease. Why would ever restricting calories make you live longer or think more clearly, be able to perform cognitively? It just seems so counterintuitive. One last thought that I want to leave you guys with before we finish this lecture is a lot of stuff seems to catch our attention, maybe seem backwards, and it's providing a lot of new ideas. And so I want to get you guys thinking on when we study the physiology of aging, we always think of a mechanism and a way to turn this gene on or off, but there's a lot of stuff that we have a hard time measuring and even have a hard time kind of believing. Do we need to change our definition of physiology in this case? And so I'm returning to one of the documentaries that we've seen a few clips from. There's one at the end that addresses the idea of mindset on aging. It looked at older men that were forced to live in a situation that was 40 or 50 years in the past. Does thinking younger and the perception of being younger, being back in time, does that change anything at the whole body physiological level? So can the mind influence the body? And it's really quite interesting. So I'll leave you with that one last clip here. This is included in the multimedia notes. It's also video 3 in the description of this video. And I'd like you guys to watch from 48 minutes 36 seconds to 55 minutes and 6 seconds. So go do that now and come back to this video just for one closing thought. So pretty amazing results from that one study. Just 1 week of study resulted in physical changes, physical improvements in aging. That piece goes on to talk a little bit more broadly about the area, and it's hard to draw hard conclusions from just this one study, but still really neat idea that the mental process of aging can affect the physical processes of aging as well. Now for this particular study, it's difficult to know whether it was the fact that they lived and thought in younger terms, or maybe it was the independence they had. It's hard to know if it was independence or if it was the fact that they were more physically active and had to move around on their own and get their own food and everything else. But still a really neat idea and a good springboard in order to explore this area further. Now that's the end of this lecture, and I will post another summary video for you guys that outlines the main points of everything we've talked about in this half of the course. So I hope you enjoyed watching. I hope you learned a little bit. I found this really exciting to talk about. And if you have any questions or comments, please don't hesitate to send me an email. Also, I'll be in my office next week, Tuesday for sure, Wednesday probably, maybe not Thursday. If you guys want to set up a meeting before the final, shoot me an email and we can set up a time, okay? All the best studying, and talk to you soon.